As you go about your day, is it ever on your mind how your body is a single entity, but at the same time, it's made up of many different interconnected systems? Or how we are miraculously able to keep ourselves in working order, despite the fact that there are so many pathogens who would love to call our body home and wreak havoc on our lives? Our immune system is tasked with keeping us healthy, but unlike our other systems, it does not have one central location. It is spread out everywhere. So how exactly do the immune cells in our body communicate with one another to maintain this protection? Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at creating an immune cell wiring diagram. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. There's also more information for you in the description below. Now before we jump right into the meat of how this diagram was created, it's important to put this information into context. The immune system utilizes both the lymphatic and circulatory systems to train and move throughout the body, allowing transport to places of injury or disease. Now, our immune system is made up of multitudes of different cells that are typically categorized based on their function, but this aligns pretty well from the type of cell they're derived from. Myeloid-derived cells are usually part of the immediate and non-specific innate immune response. These include monocytes, macrophages, neutrophils, and dendritic cells. Lymphoid-derived cells are part of the adaptive immune response, which gets activated more slowly but protects us from reinfection, making it a hallmark of our defenses. Now, these include multiple types of T cells and antibody-producing B cells. The function of each of these cell types is dependent on proteins. Proteins on the cell surface are present in unique combinations, which allows us to categorize cells based on how they appear. These surface proteins are receptors that sense things in the environment, giving the cells information on where it is and what is around it. The function of these proteins is also related to how they are shaped. Now, some of these proteins interact with other cellular proteins, allowing the cells to communicate. These dynamic connections are essential for relaying messages about where to go, what to attack and leave alone, and how to play well with others, along with the occasional gossiping about other cells. Now, 57% of receptor binding pairs are unique, with no other protein interactions. This singularity is found primarily in signaling receptors, which can tell the cell important information about the environment. To put it another way, receptors and their binding partners are kind of like puzzle pieces that fit together, where some of them only fit in one place and others can fit in multiple places. It's just that when that happens, it allows the cell to know where it is and what to do. Each of these interactions have properties that determine the strength that it binds to its target piece, and takes into account how many receptors there are to determine how strong the interaction is between cells. Adding more layers to this already complex situation, not all of the receptors interact the same way all the time, much the same as you may not act the exact same around your friends and your family. Cells possess different properties when they are relaxed, stressed, or stimulated. Now, even though lots of work has been done on identifying receptor interactions, our understanding is patchwork, as we've only identified around 140 pairs. This may sound like a lot, but there are thousands of receptors that we do not know what they bind to. Now, combining all of what we do know together, we have learned enough to adequately explain how some cell-to-cell interactions work. But there is still a long way to go before we fully understand everything. What makes studying these proteins so hard is that each of the protein conformations behaves differently, and finding a technique to analyze many different types of them can be extremely costly. Additionally, when doing experiments, you're only characterizing a small snapshot of when the cells interact. If we could get multiple snapshots, we might be able to form a sort of movie of how cells function, and be able to identify interaction patterns. This is of particular clinical importance because cell receptors can influence the cell to perform certain functions, like killing cancerous cells, or preventing overactivation of the immune system in autoimmune and inflammatory diseases, like rheumatoid arthritis or inflammatory bowel disease. Now, I want to take a moment and really highlight why cellular interaction research is so important. It is important as an end in and of itself, to gather a greater understanding of cell-to-cell interactions. In doing this exploration, though, we develop new technologies that reduce the costs and increase the effectiveness of experiments to facilitate innovation around different uses and data generation. When that happens, new technology can be used to increase the ability of researchers to accurately identify new therapies for disease. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're looking at today. This paper is called A Physical Wiring Diagram for the Human Immune System by Shilts et al. From the Wellcome Sanger Institute in Cambridge, UK. In this paper, the authors used a high-throughput screening method to systematically map most of the direct surface protein interactions of immune cells, and then modeled how these interactions would work around the body. To first do this, they had developed a method that had never successfully been accomplished before. This encompassed testing binary interactions of all possible pairings of hundreds of recombinant surface proteins to enable the systematic survey of whole cell surface protein interactions between immune cells. This method, the Scalable Arrayed Multivalent Extracellular Interaction Screen, is called Sevexis. This technique is able to screen thousands of interactions while only using a tiny amount of protein, effectively making it like a cellular puzzle piece sorting protocol. In case you were wondering the specifics, it works by exploiting protein multimerization around streptavidin, which can detect even low binding affinity interactions, so that both immobilized baits and reporter-linked preys can be easily produced from a single construct. What makes this especially impressive is that this technique enabled multiple different types of surface receptor protein conformations to be screened. By testing all possible protein pairings on the 630 proteins used, 187 positive interactions were quality checked a second time to give a comprehensive and reproducible matrix. This identified 28 never before documented interactions, expanding our collective knowledge on human immune protein interactions by 20%. To validate these discoveries, every interaction was assessed by alternative approaches, which measured the binding kinetics of each interaction. This yielded the authors with information to create a map of protein binding interactions from immune cells in the blood, where the thicker lines indicate increased binding strength. With this data, they identified that higher protein expression patterns weakly and negatively correlate with binding strength. And of note, the authors also found that higher affinity interactions predominate in an inflamed state, possibly to support the body's need for dynamic responses when sensing potential threats. Now, already the authors had validated many previous observations and generated a lot of novel information. To put this to use, they created a coarse yet principled mathematical model to try and predict immune cell interactions. This utilized multiple parameters to summarize the contributions of individual protein interactions, which then computes the overall probability of binding between two cell types. Now, any good model created should be able to make accurate predictions. And when comparing two published empirical measurements, the authors found that their model was pretty consistent. As mentioned before, immune cells move throughout the body, and each of our organs is its own specialized environment. To accommodate this, the authors sought to contextualize cell interactions across different tissues, utilizing already generated cell expression databases. This culminated in an interactive online atlas, which summarizes receptor connectivity, physical cell connections, cell type interactions, and localization of interactions amongst 8 different human tissues. This allowed the authors to determine whether immune receptor interactions proceed through shared structures or are distinct between tissues. They also identified that myeloid lineage cells, like macrophages, have higher network centrality scores despite similar protein expression to other cell types, meaning that they act as hubs across multiple cellular interaction networks. Since disease is a part of life that causes immune dysregulation, the authors also wanted to encompass this in their atlas. So they incorporated paired disease and reference samples to be able to give informed predictions on these states. Now, this is all fine and dandy, but do these predictions play out in real life? To determine this, the authors used a spatial transcriptomics dataset that gives a snapshot of the lymph node. When investigating T cells, they found that their predicted interactions are actually close to one another. This is exemplified here by the white overlapping dots of JAG1 and VAS-N. Finally, the authors pondered over the clinical significance of their wiring map and what the effects of different purified receptor proteins might have on the cells. They did this using a microscopy technique called pharmacoscopy, which images spatial patterning and direct interactions of immune cells. The authors then incubated pools of activated and non-activated human immune cells with different recombinant proteins over the course of a day. When looking at the cell interactions and activation effects at 4 and 24 hours, they were able to gather many already described and novel immune receptor interactions. Some of the interactions they were able to identify were increased monocyte contacts with SERPA and early B-cell interaction with molecules implicated in adhesion. This model works pretty well because when comparing observed to predicted cell perturbations, the computed predictions fairly accurately forecasted the actual result. Now, to quickly summarize everything all together, the authors created a method which allows them to screen hundreds of different proteins to see whether they are able to bind one another. By verifying the positive results from the screen with binding kinetic assays, the authors were able to create a receptor binding strength map. They used this information to create a computer model, which would predict different immune cell interactions and extrapolated these to different tissues. And finally followed it up with a microscopy-based technique that allowed them to verify the predicted cell interactions and the immunomodulatory effects they may have. Not only do I think that the development and verification of new tools for identifying cell interactions is exciting and important, it is also significant in a broader context. This information is significant because, well, this is the first large-scale interactive online multi-cell immune atlas. It was a massive undertaking to generate and verify all of the data. This atlas can be used to help others further refine their experiments and drive hypotheses of how cells might interact with biological agents. And with greater predictive power, this increases the capacity for identifying new and effective therapeutic agents that could alleviate disease. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. It is great to have developed an immune cell interaction atlas, but every day our knowledge of how these interactions happen is getting broader. The authors have their code available for others to see and use, but is there a way for this information to auto-update itself as new information comes along? Additionally, can this information be integrated with protein modeling software to predict entirely novel protein interactions? When looking at Cevexes as a technique, is it limited to just looking at immune cells at rest? Or can it be used during novel infectious outbreaks like COVID or monkeypox to get a better idea on exactly how the pathogenic proteins are altering cell populations, leading to an effective therapy? Additionally, can this technology be used for personalized medicine, where perhaps tumor samples can be analyzed and assessed to see which therapies would be most effective for individuals? I mean, hopefully. As always, though, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope you enjoyed your time doing so. If you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.